Have you ever wished you could edit a book, fixing a typo with just a few keystrokes? What if I told you that scientists can now do something similar, but with the book of life itself, our DNA? This isn't science fiction, it's the reality of CRISPR, a revolutionary technology that's changing the world as we know it. Today, we're going to dive deep into what CRISPR is, how it works, and why it holds the power to reshape our future. So what exactly is this thing with the catchy name? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. I know it's a mouthful, but the core idea is surprisingly simple. Think of it as a biological search and replace tool. It was first discovered in bacteria, where it acts as a primitive immune system. When a virus attacks a bacterium, the bacterium captures a small snippet of the virus's DNA and stores it in its own genetic code. Within these CRISPR regions, it's like taking a mugshot of the enemy. If that same virus ever returns, the bacterium is ready. It produces a special RNA molecular copy of that viral mugshot. The real breakthrough came when scientists, including the Nobel Prize-winning duo Emmanuelle Charpentier and Jennifer Doudna, realized this bacterial defense system could be repurposed. They figured out that they could design their own guide RNA to target virtually any gene in any organism, from a plant to a mouse, and even to a human. This meant we could now find a specific DNA sequence and cut it with incredible precision. But what happens after the cut? That's where the magic really begins. When our cell's DNA is cut, they have natural repair mechanisms to fix the break. The cell can either try to patch the two ends back together, which often introduces small errors and can be used to knock out or disable a gene. Or if scientists provide a new piece of DNA as a template, the cell can use it to repair the break, effectively pasting in a new, desired sequence. This is gene editing, the ability to delete, repair, or replace genes. Let's make this more concrete. Imagine a person with a genetic disease like sickle cell anemia. This disease is caused by a single tiny typo in a single gene. With CRISPR, doctors can take a patient's blood stem cells, use the CRISPR-Cas9 system to find that specific typo, cut it out, and let the cell's repair machinery fix it using a correct template. These edited, healthy cells can then be returned to the patient, where they can produce normal blood cells. This isn't just a theory. Clinical trials are already showing incredible promise, with some patients becoming essentially free of the disease's debilitating symptoms. The potential applications are mind-boggling and extend far beyond human medicine. In agriculture, scientists are using CRISPR to create crops that are more nutritious, resistant to drought and pests, and have a longer shelf life. Imagine tomatoes that stay fresh for weeks, or wheat that can grow in salty soil. This could be a game changer for global food security, helping us feed a growing population in the face of climate change. CRISPR is also being used to create allergy-free peanuts and decaffeinated coffee beans. The possibilities are nearly endless. In the field of conservation, CRISPR could even help us bring back extinct species, a concept known as de-extinction. Scientists at organizations like Colossal Biosciences are working to edit the DNA of an Asian elephant, which is the closest living relative of the woolly mammoth, to reintroduce mammoth-like traits. Their goal is to create a functional cold-resistant elephant that could one day roam the Arctic tundra, potentially helping to restore that fragile ecosystem. While it's a complex and controversial idea, it highlights the sheer power of this technology. Of course, with such immense power comes immense responsibility. The ability to edit the very code of life raises profound ethical questions. One of the biggest debates revolves around germline editing. This means making changes to the DNA of sperm, eggs, or embryos, changes that would not only affect the individual, but would also be passed down to all future generations. While this could potentially eradicate hereditary diseases forever, it also opens a Pandora's box of concerns. Where do we draw the line between therapy and enhancement? Is it okay to eliminate a disease gene? What about editing genes for height, intelligence, or athletic ability? This leads us down the slippery slope of designer babies. creating a world where genetic modifications could deepen social inequalities. Who would have access to these expensive technologies? Would we create a new form of genetic divide between the haves and the have-nots? These are not easy questions, and society needs to have a broad and inclusive conversation about the moral and ethical boundaries we should set. There are also safety concerns. While CRISPR is incredibly precise, it's not perfect. There's a risk of off-target effects, where the Cas9 protein might cut the DNA in the wrong place, leading to unintended and potentially harmful mutations. Researchers are working tirelessly to improve the accuracy and safety of CRISPR systems, developing new versions like prime editing and base editing that offer even greater precision with fewer risks. But the technology is still evolving, and we must proceed with caution, especially when it comes to human applications. The story of CRISPR is a testament to the beauty of basic scientific research. A discovery made while studying the humble immune system of bacteria has unlocked one of the most powerful tools in the history of biology. It gives us an unprecedented ability to understand and rewrite the script of life. It offers hope for curing devastating genetic diseases, creating a more sustainable food supply, and even protecting our planet's biodiversity. But CRISPR is not a magic wand. It is a powerful tool, and like any tool, its impact will depend on how we choose to use it. It forces us to confront fundamental questions about what it means to be human and what kind of future we want to build. As this technology continues to advance at a breathtaking pace, it's crucial for all of us, not just scientists and policymakers, to stay informed, engage in the conversation, and help shape the future of gene editing responsibly. CRISPR is more than just a scientific acronym. It's a new chapter in the story of life itself, and we are the ones who get to help write it. Thanks for watching, and if you found this journey into the world of gene editing fascinating, be sure to like this video and subscribe for more deep dives into the science that is shaping our world. See you next time.